in this, I will be discussing about the understanding the basic flow to yeast species identification. So first, we should know which whenever yeast colony has been grown, we have to keep in mind these options. The yeast are either Basidiomycetes yeast or Acamycetes. So in the Basidiomycetes, following species are there: Cryptococcus species, then Trichosporon, Geotrichum, Saccharomyces, and Magnusiomyces species. These are included in the Basidiomycetes yeast, and Acamycetes yeast, in that Candida species Saccharomyces anomalus, which was formerly known as Pichia anomala and Hansenula anomala. Then Malassezia species, Rhodotorula species, Sporobolomyces salmonicola species, then Saccharomyces species, Kodamia omeri species, and Magnusiomyces species, which was formerly called as Pseudozyma. So whenever when I was new, I used to think that what any yeast colony has come, it was a I was thinking it is some type of Candida species, but there are so many species which will give yeast like growth. So we have to do differential with that. So for approaching the diagnosis of yeast isolate, we are having conventional methods, automated identification systems, and molecular methods. So in conventional methods, we will do gram stain and India wet mount first. Then we will go for the India germ tube test, Dalmau method for the on the corneal agar we will do. Then chrome agar Candida medium for differentiation of each species. Different species will give different colors on the chrome agar. Then urease test. So some yeast are urease positive. I will discuss later. So urease test is important. Then sugar fermentation test, sugar assimilation tests, temperature studies, and acetopore formation and detection test. These will come under the conventional methods. And we are having automated identification systems which are based on the biochemical reactions. So these are Vitek 2 system, API 20 CAUX system, API Candida system, BD Phoenix system, and Microscan Beckman Coulter system. These are automated identification systems which identifies on the basis of biochemical reactions. Then some molecular methods like Maldita of MS method, PCR, and sequence-based method. So whenever we need to identify any yeast isolate, first we have to check for the purity, whether it is mixed or not. So for checking the purity, we can do either direct microscopy with LPCB and check whether any mixed growth is there or not. Then we can substitute it on the chrome agar and check for the color change. And we can do on the SDA plate for checking the different morphology. So we should not get the mixed cultures. That's why for prevention of contamination, we will use the antibiotics in the media. For bacterial contamination, we will use antibiotics, and for saprophytic fungi prevention, we will use cycloheximide. And if there is a mold contamination of the yeast colony, then we will give isolation on the yeast malt agar or isolation on the yeast malt bath. In this only yeast colonies will be grow. Whenever any yeast isolate is grown, so gross cultural characteristics should be seen first, and then we will go for the microscopy. So gross cultural characteristics, we can see on the SD here. You can see moist circular yeasty pasty colonies are there. So this is one morphotype. Another is these are dry, rough, dull yeast colonies. So they will give a clue like dry, rough, dull colonies can give diagnosis. Means we can suspect Trichosporon, Geotrichum, like that. And if there is any specific pigmentation, like means orange or coral red pigment is there, then it will be suggestive of different species. So grass cultural characteristic is important. Afterwards, we will go for the microscopy. In the microscopy, actually, we have to see the asexual structures like blastospores, pseudohyphae, true hyphae, then arthrospores, chlamydospores. Presence or absence of these things will decide the species. And type of budding should be noted, like which is unipolar, multipolar, or by fission. And shape and the size of the cells are very important. So these are about the asexual structures. So in Accelerate family, we will get the azospores and basidospores also. So sexual structures, arrangement, cell wall, number, shape, and size also matters. According to these microscopic characters and grass features, it will give clue for the species identification, and we have to do the carbohydrate assimilation and fermentation tests and other conventional tests for detecting the species. So first I will now I will be describing one by one these yeast species, how they will look and how to identify them. So Candida species, around 200 anamorphic species are there, and around 13 phylomorphic genera are there. So out of them, around 20 species are the significant pathogens. So some important Candida species which are newly renamed are Pichia pudrivazia. That is, formerly it was called as Candida krusei. Merozyma gulurmandi, which was formerly called as Candida gulurmandi. Clavispora lusitani. It is now, but formerly it was called as Candida lusitani. Then Cluveromyces marxinianus. Formerly it was called as Candida caper. And Bicterhamomyces anomalus. Formerly it was called as Candida pelliculosa. Candida albicans complex, Candida glabrata complex, and Candida paraxylus complex. These are renamed. So for the identification of Candida species, conventional methods, by conventional methods following what we can do, like urease test positive Candida or Candida krusei and lipolytica. Germ tube test is positive in the Candida albicans and dublinensis. Then esculin hydrolysis is positive in Candida norwegensis. Then rapid assimilation of Trehalose test is positive in Candida glabrata. So with the help of the conventional methods, we have to identify between the Candida dublinensis versus Candida albicans, Candida gulurmandi versus Candida famata, Candida krusei versus B capitis, Candida krusei versus C, Candida inconspicuo versus Candida norwegensis. So these can be identified with the help of conventional methods. Candida we have to identify Candida glabrata versus Candida auris versus Candida humulini. So in the gram stain, yesterday I have shown you these pictures. So in the gram stain, whenever there is any yeasty colony comes, first you check for the purity and you do the gram stain for whether it is a pure colony or mixed, and how the structure looks. Like here you can see B by C with pseudohyphae. Here also pseudohyphae, circular B by C. So, and Candida krusei will give the elongated yeast cells. Yeah, and Candida auris, the size, they will be only budding cells, but they are smaller in size than the glabrata. So, on Dalma plate method, we will see certain morphological characters and we will decide the species. Here you can see this terminal chlamydospore. So, it is Candida albicans. So if the chlamydospores are there in the groups, then it is called as Candida dublinensis. Then and we have to differentiate by other conventional methods in between these two. If this chlamydospore formation if we will do for the means after 48 hours, more incubation is there, 3 to 4 days means these terminal spores will change into the groups. So for differentiating these two, we have to do other conventional methods also. So this is the dermaplate method. Here you can see microscopic appearance of C. tropicalis, means long pseudo hyphae along with teardrop shaped, and it looks like a lace-like appearance. This is a Candida tropicalis with long pseudo hyphae. Here, this is Candida paraxyllosis. These are curved pseudo hyphae, and curved and short pseudo hyphae will be there, and there will be oval blastoconidia are there that can have the multilateral budding also. This is Candida glabrata. Here only yeast cells will be there, circular to oval yeast cells without any pseudo hyphae. And this is Candida humulini. Here also the only yeast cells will be there. There won't be any pseudo hyphae. So whenever any species will come which will give the on clone mill agar only budding yeast cells, no pseudo hyphae, then we have to label the species as Candida glabrata versus Aris versus humulini, or Aris or humulini. So it cannot be differentiated by routine conventional methods. For Candida glabrata, at least we can exclude by rapid assimilation of pre-Harless test. It is given positive by only Candida glabrata, and by White identification system also we can label it as glabrata. But Candida auris versus humulini, there actually we have to do by molecular methods only. So Candida auris is phylogenetically closely related to the Candida humulini species complex. So hence the Candida auris is misidentified as Candida humulini on the White system. So the confirmation by the molecular methods is necessary, like mallet of MS, DNA sequencing and PCR can detect Candida auris. So this is one paper. In this, actually, the misidentification of Candida auris by different diagnostic methods means they have evaluated. So different systems, identification system, it is depend on the biochemical reactions. Hence, these identification systems will give the Candida humulini or other species as a Candida auris cannot be detected. Their database is not having that species. So in the white type, the Candida auris is misidentified as humulini or Lusitania or Famata. So and the API misidentified the Candida auris as C. famata, API 20 CAUX system as Rhodotorula or unidentified. It will give Candida auris. So microscan Beckman culture, it will misidentify Candida auris as C. famata, Lusitania, Guillermandi, Paraxyllosis albicans or tropicalis, anything. So in this paper was in 2018. In this mallet of MS also misidentified C. auris as albicans or humulini, but now currently the database of mallet of MS has been updated, so it can identify C. auris correctly. So for the correct identification of C. auris, the best methods are first mallet of MS at present, and then C. auris specific PCR and DNA sequencing. So I want to share with you this. This is an equal Candida score 2018. So an ECMM score derived from current guidelines to measure the quality of clinical management of Candida. So this is Candida score. This is something different I found. In this, actually, the equal Candida score weighs and aggregates the factors recommended for the ideal management of Candidemia and provides a tool for antifungal stewardship as well as for measuring guidelines adherence. Current guidelines provided by the European Society for Clinical Microbiology and Infectious Diseases and by the Infectious Disease Society of America, they reviewed, and the strongest recommendations for management authorities selected as the basis for the scoring score. So this scoring will be actually in two types, non-CPC carriers and in the central venous catheter carriers. Two different scores are there. It depends on the diagnosis, treatment, and follow-up. All the three things should be checked. So in the diagnosis, in this equal Candida score, in the diagnosis, we will see about the initial blood culture, species identification, susceptibility testing, echocardiography, and ophthalmoscopy. Whether all these things have been done or not, we have to check, and according to that, we have to score. In the treatment part, ecocandin treatment, step down to fluconazole depending on the susceptibility results. Treatment for 14 days after first negative follow-up culture. And CVC carriers, in this CVC removal, these factors are there. And in follow-up, whether we have done follow-up blood culture or not, these all the components will be there in the Candida score, and it is very helpful in the clinical Candida management, which will be measuring the quality of Candidemia management. So next, this was all about the Candida. Now we will move on to the another species now. So Vicarihamomyces anomalus. This was previously Candida pelliculosa. In the sexual state, it was called as Pichia anomala and Hansenula anomala. In asexual state, it was called as Candida pelliculosa. So the grass morphology, it will be smooth, moist, cream-colored yeasty cells. And in the microscopy, we can see the in the asexual state, we will see ellipsoid budding yeast cells. Ellipsoid budding yeast cells with sometimes pseudomycelium. And in the sexual states, we can see the ascospores. The ascus will be the helmet or hat appearance, and they will be homothalic. So this is these are the colonies of Vicarihamomyces anomalus, yeasty colonies, and we can see here the ellipsoid budding yeast cells, sometimes with pseudomycelium, and these are the ascospores we can see. So next species is Kodamia omeri. This species is commonly encountered in the Fungemia immunocompromised patient. So this is actually grass in the calf morphology, cream colored, slightly ramified by the pseudohyphae. So cream colored, this is a reverse surface, this is a upward surface. So here we can see in the asexual state, budding yeast cells in single pairs or short chains, and there will be a pseudohyphae. And in sexual stage, we can have the hat-shaped or spherical ascus. So budding yeast cells will be there, which can be in the pairs or short chains, and pseudohyphae can also be there. So next species is Cryptococcus species. In the Cryptococcus species, we will differentiate between the neoformans, var neoformans, and var grubi or Cryptococcus gattii. So on the grass morphology, they will be cream colored at first, and later become the tannish, flat or slightly hipped, shiny, moist or mucoid colonies will be there. So in the microscopy, round dark walled budding yeast cells will be there, and in indaying wet mount for capsule, it will be positive. In the sexual state, it is a Basidiomycetes species. So basidium will be there in the sexual state. Upon mating, dikaryotic hyphae with clamp connections are formed, bearing slender hyphae, and percepted basidia with terminal swelling produce spherical basidiospores. So microscopy here you can see this is the basidio clamped hyphae with terminal basidio spore heads. So this is basidium, and these are the conidia formed. This is the sexual state of Cryptococcus. So this was about the microscopy, and it will be giving growth on the birdseed agar or nightseed agar, brown colonies it will be giving, and all species of the Cryptococcus lack fermentative ability. Hence, the sugar fermentation and assimilation tests are not useful in speciation of Cryptococcus, and all Cryptococcus are urease test positive. So to differentiate between the Cryptococcus neoformans gattii to the other Cryptococcus species, we have to do the caffeic acid disc test. So it will be positive by only these two, Cryptococcus neoformans and gattii, and growth at 37 will be positive, will be given by only these two species. Other species of Cryptococcus won't be grown at 37 degrees Celsius. So this test will differentiate whether it is neoformans and Gatti. Now we have to check for the Gatti and neoformans. So for that we use CGB agar. In that, Gatti will grow and turns the agar blue, cobalt blue, and neoformans won't. So after that, we have to check for the neoformans, war neoformans versus groupie. For that, we should use CDBT agar, in which the neoformans will change orange color growth, and groupie will not will give no growth. So here you can see this is the birdseed agar having brown colonies of Cryptococcus, and here this India ink with capsule. On the gram strain, circular spherical budding is sets we can see, and this is the sexual state of Cryptococcus species, where the basidium is there, conidia are there, and clamp connections are there. Terminal basidiospores are there. So, like Candida score, here we can have the Cryptococcus score, equal Cryptococcus score. This is a European Confederation of Medical Mycology score derived from current guidelines to measure the quality of clinical Cryptococcus management. So, the equal Cryptococcus score weighs and aggregates factors for ideal management of cryptococcal infections. So, equal scores reflects the strongest recommendations from the current guidelines. The scorecards are quick reference to measure the guideline adherence to the and support the antifungal stewardship. In this diagnosis and management part will be included in the score. That's antifungal, which drug we are using, immunomodulation, ID consult, and follow-up. These components will form the part of the score, and according to the it will help in the assessment of adherence to the treatment or to the guidelines and to support the antifungal stewardship. So now we will move on to next species, Malassezia species. This is a lipophilic unipolar yeast. So the different species are Malassezia Malassezia pachydermatis, Malassezia sympodialis, Malassezia globosa, restricta, obtusa, and sloophy. So on the microscopy, clusters of yeast cells will be there, and short and curved blunt hyphae can be there. For the growth of the species, it cannot go on the SDA. We have to use special media, that is modified Dixon medium, Liming and Ottman medium, and Dixit medium, and modified chrome agar medium. So sugar fermentation and assimilation tests are not applicable. We cannot use this test for the differentiation of Malassezia species. So here in this microscopy, you can see the budding yeast cells with short pseudo hyphae, typical of this Malassezia species, and these are the colonies on the modified Dixon agar of Malassezia species. Next species is Trichosporon species. So important species are Trichosporon asahi, inkin, fecal, and asteroids. For the presentive identification of the Trichosporon species, they will be forming the dry colonies, dry yeast colonies on the SDA. Then we have to put the urease. If the urease is positive, and on microscopy, if we can see the arthrospores along with the blastoconidia, then it indicates towards the Trichosporon species. Urease positive, giving on grass appearance the dry colonies, and in microscopy it will give arthrospores plus arthroconidia, blastoconidia and arthroconidia. So on the commercial biochemical methods, it cannot be differentiated because they are non-fermentative. So these commercial methods will be based on the biochemical test identification. So on the Vitek and other instruments, it will give either Trichosporon asahi and non-asahi. but it cannot differentiate between the individual species. For that, we have to do the molecular level detection. We have to do by multi-pass MS or IGS1 region sequencing. So there are some global guidelines for the diagnosis and management of rare infections, which is an initiative of the ECMM in cooperation with ISHAM and ASM. These are recent guidelines, 2021. So in these guidelines, there is a management of rare yeast. They have and detection, diagnosis and management of rare yeast. They have given these guidelines. So in this invasive infection due to Trichosporon species, we have the clinical samples will be blood cultures, biopsy tissue, urine, and other body sites. Meticulous history and physical examination is warranted to find the site of infection. Then we should do the culture. On culture, gross morphology, we will get yeast colonies, which will be dry, and in the microscopy, we will get pseudohyphae, or arthroconidea will be there. So then we have to do for other identification tests, microscopic morphology and urease test. If urease test is positive, we have to give it as a presumptive Trichosporon species, and then we have to go for the different methods for the detection, like multi-top or biochemical tests. But the means conventional most of the identification systems cannot give the correct species of Trichosporon. So Maltitop and the gene sequencing IGS1 from the ribosomal DNA sequencing will be needed for definitive species identification. And after that, we should perform the antifungal susceptibility testing according to CLSI guidelines. So these guidelines are given by ECMM in cooperation with ISHAM and ASM. So next, Geotrichum species is closely related to the genus Saccharomyces and Magnusiomyces. In this only Geotrichum candidum species is pathogenic. So the presumptive identification for this will be cross morphology. It will be forming dry, white to cream, smooth or hairy colonies. Then it will be urease negative, but it will be having the presence of arthroconidea and absence of plastoconidia. It will be in the carbohydrate sugar fermentation tests or assimilation test. Actually, assimilation of glucose, galactose, lactose, and xylose will be there, but saccharose and maltose will be negative. So again, for this Geotrichum, the 2021 guidelines from ECMM in cooperation with ISHAM have been released. For that, actually, clinical samples will be blood culture, biopsy, tissue, urine, and other body sites. Meticulous history and physical examination is warranted to find the site of infection. Then we will go for the culture. There we will get the white, dry, fluffy colonies within 48 hours. So, and in histopathology, septate hyphae can be seen. Concomitant tests for identification we should perform, and on the morphology, microscopic morphology, hyphae with cylinder-shaped arthroconidea and no blastoconidia will be there. And on the chroma candida, will give pink color colonies, which might suggest of Geotrichum. And we have to establish the Geotrichum species infection and perform other tests like multitop and fungal PCR followed by DNA sequencing for correct identification of the species. And we have to perform the antifungal susceptibility testing according to CLSI guidelines. So next species, Magnusiomyces species. It is actually previously known as Geotrichum capitatum or Blastoschizomyces capitatum. Now it is called as Magnusiomyces capitatus. So the gross morphology, it will be white to cream yeast-like, smooth to wrinkled, developing short aerial hyphae with edge, radiating edges. So in the microscopy, there will be round to oval budding cells, hyphae, pseudohyphae, and few arthroconidea. Arthroconidea and many aniloconidia will be there, and annelids will be formed. So the identification by biochemical methods is not possible for this species. We have to go for the molecular or molecular tests. other molecular tests, DNA sequencing and PCR. So here you can see these are the plasma yeast colonies of Magnusiomyces capitatus, and here you can see the Aneloconidia and Annelids. So next species is Saccharomyces species. This also is the emerging cause of fungemia. So actually this species differs from Magnusiomyces capitatus by lacking sympodial conidiogenesis and being cellobiose-salicine positive, but may not coincide with the molecular differences. Means Magnusiomyces capitatus and Saccharomyces, they are closely related. So only molecular identification, DNA sequencing will be used for the correct species identification. Next is Rhodotorula species. The most commonly encountered species are Rhodotorula, mucilaginosa, glutenis, and minuta. In the gross morphology, there will be coral red to pink color pigment, or there will be glistening, smooth or rough, and soft colonies. So this is it is due to the pigment carotenoid. So coral red to pink colonies will be there. On the microscopy, oval or elongated budding yeast cells will be there, and few rudimentary pseudohyphae. In the Rhodotorula species, how we will diagnose first with the color of the pigment, and then only BYC will be seen, and we can do identification by the sugar assimilation and fermentation tests. So hence, Vitek2 system will give correct diagnosis of Rhodotorula, and molecular methods also can be used for diagnosis. So similar to this, one of the species is there which will give coral red to pink color colonies. So this Porobolomyces salmonicolor. Previously, I used to think that if the pink pigment has come, orange pigment has come, it is only Rhodotorula. But now it has. Now I understood that if this type of colonies has come, there are two differentials: Rhodotorula species and Porobolomyces salmonicolor species. So, but in this the microscopy, only budding cells will be there. Here we can see the Ballistoconidia. Means in this satellite colonies can be formed due to the production of this is in the microscopy, oval to elongate yeast-like cells will be there. Pseudohyphae will be there. Pseudohyphae will be there, and true hyphae will be there, and kidney-shaped Ballistoconidia, which were supported on the denticles. These will be there in the microscopy. So if this type of colony has come, if it is budding its cells, then it is going towards the Rhodotorula. But if it is giving rough structures like this, having true hyphae, pseudohyphae with Ballistoconidia on denticles, then it will go towards the Porobolomyces salmonicolor. So to identify this species, what we have to do on the cornmeal agar, we have to inoculate these colonies, and on the opposite side of the cornmeal agar, we have to put another cornmeal agar plate. So they will be attached to each other, and we have to keep in the incubator. Both the surfaces will be facing each other, and the inoculated media will be on the upper side. So if the Ballistoconidia are formed, they will be shooted, and they will produce a satellite colonies. So on the downside surface of the cornmeal agar, there will be two surfaces. Upper surface where we have inoculated, there the colonies will be developed on the next day, yeasty colonies. But due to the production of Ballistoconidia, they will shoot up to the another agar. So down agar also will be having satellite colonies. So it will be indicative of Porobolomyces salmonicolor species. And this test will help to differentiate between Porobolomyces and Rhodotorula. So here you can see this is actually yeasty colony. Here Ballistoconidia are there, true hyphae, pseudohyphae. We can see this is denticle, and this Ballistoconidia is there. So next species is Saccharomyces cerevisiae. It is called as baker's or brewer's yeast. So it is responsible for fungemia, which is attributed to the uses of prebiotics. So on the gross morphology, it will give white to cream color, moist and smooth colonies. On the microscopy, round or oval budding is cells with very few short pseudohyphae we can see, and sexual state of this will produce the ascus like this. So this is Saccharomyces cerevisiae. Here you can see budding is cells, round or oval budding is cells with multilateral budding. Here multilateral budding can be there with very few short pseudohyphae. Next species Magnusiomyces, which is previously called as pseudozyma. In these two species are there, M. affidis and M. pullatus. In the grass morphology, it will be flat, velvety, whitish to pale yellowish cream, zonate towards the margin, and margin is fringed. So here, Magnusiomyces species. In the microscopy, we can see aerial mycelium consists of branched acropetal chains of fusiform conidia. For the conidia will be fusiform, and hyphae will be abundant with retracting septa in cells separated by the lyse cells. Hyphal cells forming the blastic conidia on denticles or short stegmata. These conidia will be hyaline and fusiform, and budding cells often contain oil droplets. Here you can see oil droplets also you can see. So this was about the Magnusiomyces. Lastly, I will talk about Prototheca species. Actually, it is not a fungus. That is this is a chlorophyllous algae, which will give yeast-like colony on the Saccharomyces dextrosaga. So the important species in humans is P. wickerhami. Other species are P. zophi and stagnora. On the grass morphology, they will give dull white to cream color yeast-like colonies they will give. So on microscopy, you can see the we can get the sporangia containing sporangia spores, and there will be no budding cells or no hyphae will be there. So whenever any yeast colony has come, we have to think about all these different shells for the correct identification. So thank you. Now now we will show one video for yeast identification flow. So Karuna madam.
Yes madam, we are ready.
Ma'am, okay. Please play the video.
Yes, sure.
Good evening to all. This is Dr. Anish, resident from the Department of Microbiology. Today I present the identification flowchart for yeast. The identification of a yeast isolate begins with a gram stain. The gram stain that we follow is a modified version of Hacker's. The next step that we have is an India ink preparation. For India ink preparation, we use the India ink. A clean slide is taken and a flamed cover slip is taken. An isolate is mixed with 1 drop of India ink on the clean slide and a cover slip placed and observed under the microscope. Next step that can be performed is the LPCB direct mount. A lactophenol cotton blue direct mount is performed to study the yeast morphology, microscopic morphology, and to determine the purity of isolates. Once the capsulated yeast are ruled out and the isolate is confirmed to be pure, the next step in identification in yeast is the germ tube test. The germ tube test is used for presumptive identification of Candida albicans and is a rapid screening test. But this test must be validated with cornmeal agar. We make a very light suspension of the test organism in 0.5 ml of sterile pooled human serum and incubate at 37 degrees Celsius for exactly 2 hours. Then, after incubation, place one drop from the incubated serum on a slide with a cover slip and observe under the microscope for production of germ tubes that represent initiation of hyphal growth arising directly from the yeast cell. Please note that the germ tubes will be having parallel walls at their point of origin and are not constricted. And to record a positive, about 30% of the cells should show germ tube production. Also, controls should be used. A non-strain of Candida albicans as positive control and Candida tropicalis as negative control. The medium, the inoculum size, the temperature of incubation, the concentration of the simple carbohydrates and microaerobic conditions all can influence the GT formation. Also, remember that almost 95% of clinical isolates of Candida albicans can produce germ tubes when incubated in serum. The next test after germ tube test is the Dalmo plate, and it is performed on corn meal agar containing 1% tween 80, usually in a 90 mm plate. We divide the plate into 4 quadrants and label each quadrant. Using a sterile needle or straight wire, lightly touch the yeast colony and then make 2 to 3 streaks. Place a flame sterilized and cooled 22 mm square cover glass over the control part of the streak. Thus, it provides a partially anaerobic environment at the margins. The plates are incubated at 25 degrees Celsius for 3 to 5 days. Remove the lid of the Petri plate and observe under the light microscope to observe morphological features like hyphae, pseudohyphae, chlamydospores, etc. After getting a preliminary idea about the yeast from the cornmeal agar, next test that can be performed is the chrome agar. The chrome agar is a chromogenic agar which helps us to determine the color, the species of the Candida with the help of the color that is formed on the media. The next test that can be used is certain biochemical reactions. The first biochemical reaction that we describe today is the urease test. The urease test is performed on Christensen's urease media, and the isolate is streaked onto urease agar and incubated at 25 degrees Celsius. Controls should be used in this stage also. Usually, Candida albicans used as a negative control and Cryptococcus neoformans can be used as a positive control. Sugar fermentation. Liquid fermentation media. Filter sterilized sugar at 2% concentrations added to this. This is taken in the volume of 5 ml and a sterile Durham's tube is put into each tube. And heavy inoculum is prepared from a media which is sugar free. Hence, we may remember that nutrient agar can be used for subculturing the isolates for sugar fermentation test. Inoculate each carbohydrate broth with approximately 0.1 ml of the inoculum and incubate the tubes at 25 degrees Celsius up to 1 week. Observations of the tubes must be made every 48 to 72 hours. For production of gas, please note that production of gas indicates fermentation. Next test is the sugar assimilation test, for which we follow the disc impregnation pour plate oxanographic method of Wickerham. For this, we need to prepare the yeast nitrogen broth 2 ml and separately and sterilize by filtration and can be stored at 4 degrees Celsius. Agar is prepared in 18 ml quantities and stored separately after autoclaving at 4 degrees Celsius. On the day of the procedure, the yeast suspension from a 24 to 48 hours old culture is prepared in 2 ml of YNB by adding a heavy inoculum and this suspension is added to 18 ml of molten agar, that is cooled to 45 degrees Celsius and mixed well. Pour the entire volume into a 90 mm Petri plate. Once the Petri plate is set to room temperature, the agar surface hardens. After the agar surface hardens, place a carbohydrate impregnated disc on the agar plate. The preparation of the disc can be done in-house or obtained commercially. If done in-house, sterile discs are used and 10% filtered sterilized sugar solutions are added to it. After drying the disc, they are stored at 4 degrees Celsius. The plates are incubated at 37 degrees Celsius for 3 to 4 days. The presence of growth around the disc is considered as positive for that particular carbohydrate. Growth around glucose disc is recorded first, which serves as a positive control, indicates the viability of the yeast. And here we may remember that 6 sugars are used for sugar fermentation and 12 sugars are done for sugar assimilation. So with this we come to the end of the flow chart for the identification of yeast. Thank you.
In this video, we have tried to show the yeast identification flow. If there are any doubts, you can mail me on this